Yes, ma'am. So also, please don't forget that Fallon's lab study session is tonight at 7 for the lab final.
That's right, I totally forgot about that. So that's brand new. So I lied, I did have one announcement. And what time did you say that was at?
7
7,
p.m.
right? So this is new, it's in an unusual time slot. We've not done it before, and she just sort of opted to do it because there was so much request for it. So do keep in mind that Fallon is offering to run a lab study session. Don't forget that we do have our lab final the week after we come back. It's just one of those awkward things. You don't do lab tests during finals week. It's always the last week of classes, which is now weirdly hedge-pinned as this one lone week right after a break. So try to keep that in mind. This is a good tool that we've not been able to do before. So special thanks to Fallon for setting that up. And thank you for reminding me, Miss Lafferty. Okay. When we left off last time, we were talking about our support enzymes, right? That specifically helped us with stabilization. Today is going to be the day. It's my last chance, right? So remember that these enzymes, they do not actually do any nucleotide laying. Okay, so they're not in the replication bubble laying down RNA or DNA. Their big goal is to make sure that our double-stranded DNA is opened and it's not going to bend, fold, and twist inappropriately. Right? So they're basically prepping the grounds that we're going to lay new tile on. Okay, so they're like our demo crew if we're considering this as a construction site, right? Okay, so we talked through, I think we finished this. We did, but I'm going to just do a slight review. We talked through all three of these and how they each had unique roles to making sure this goes well. So remember, we thought of helicase as our zipper. This is actually doing the pulling apart of the connected double strands into two separate single strands. All right, so there's our helicase. Okay, add these replication forks, right, unzipping. Okay, then our single-stranded binding proteins. Okay, big name, but at least they tell us what they do. Okay, this isn't actually showing us very well what it is on here, but remember, we want to think about our DNA kind of being like tape. And so once we unzip it, just like when you pull tape off its roll, it kind of wants to stick back down on itself, which is super annoying. So if we want to get in there and do that duplication process, we want to keep them apart. So our single-stranded binding proteins, I'm going to keep drawing them in orange now, are going to come in, okay, and bind to a single strand, as the name implies for us. Okay, along the way here, until our workers from the previous slide, right, our primase and our polymerases, can come through. It's basically going to keep them from sticking back together. Kind of like when you fold over the corner of the tape, right? Can't stick anymore. All right, and lastly, our class of All right, so these lessen what's called torsion. That helps you, topoisomerase, torsion. Otherwise, it can be a lot of new words, right? So this is that double twisting. You kind of like the power cord to your computer, and you toss in your bag, and you pull it back out, and it always seems like quadruple kinked. You say, how? How did you do that in the bag in 2 seconds, right? So you kind of have to do the foop, foop, foop motion before you plug it in, right? It'll get kinked so tight, the plastic around your cord starts to crack. We've all seen that with really old cords, right? We don't want that to kink so tight it starts to crack and break. So we need someone to come through and untwist that, right? So that's these guys. As we go to pull this apart, we need to untwist all that obnoxious kinking and twirling that it's going to do. These are our topoisomerases, right? Gyrases, anything that's going to do twisting. Okay, this is where we left off last class. Wanted to make sure we didn't rush through it. Okay, so this is our support staff, if you will. Any questions about anything here? Okay, and the role that these types of enzymes, remember, right here we have that ASZ ending. So what kind of biomolecule are these? Proteins. Good. All right, excellent. Okay, and as always, right, nice big, if we think about drawing it like we've been doing before, this is like half a replication bubble. So I've got my dark blue here as my DNA temp. Made it 10 minutes before doing it, right? As my DNA template strand. That's in there. I grabbed red. I'm all flustered now. Okay, so here we see helicase doing that unzipping. Okay, so it's right there at the replication fork. Okay, opening that up. Our single-stranded binding proteins are coming in right behind it. Okay, making sure that as we open that up, that they don't instantly stick back down together. Okay, and then away from all of that is our topoisomerase, right? Because its goal is just to make sure that the whole line, our power cord, right, isn't getting kinked, twisted, and bent. We see that all these guys are separate, okay, from things like primase, right, which is going to be laying down that 5' cap, right, our runway. Or polymerase, which would then come through and lay down, right, our DNA nucleotide. So none of these guys are doing any of that business. This is all separate support. Yes, and that's the next thing we're going to talk about. Perfect lead-in. Sorry. No, never be sorry. Everything up with that. Luckily, it never seems to forget me. We can't do this one yet. We'll come back to this. Boldface lie. Okay, so the last thing we're going to talk about, we're still going to do the Menti, don't worry, we're just going to do it at the end, is, and if you were sitting in here, we just had a question about Okazaki fragments. So the very last thing I want to talk about is when we're actually doing DNA replication, we have two different builds going on, what we call the leading and the lagging strand. And this develops because of that one rule we have, that we have a 5' and a 3' end, and we can only build on that 3' end. So let's look at this problem in detail. All right, so let's just look at the top. Okay, so here's our familiar territory. Rude. Okay, so this is the stuff that we know. We actually know everything on here. We just need to think about it. So we know that DNA polymerase and all of the building enzymes only synthesize in one direction. Okay, this is one of the big rules that we focused on, right? We're only adding on that 3' end. Okay, remember we talked about this being like Legos, right? We only can add on that open end, right? We can only put them on the bottom. This is a big rule. So everything that we've done up to this point, conceptually, has been creating what we call the leading strand. Okay, so we're just putting a term on it. Okay, which is the strand that has that 3' end, that building end, heading towards our replication fork. Okay, so we're going to picture the one that's in the box on my image here. So here's my primer. All right, my 5' cap, that's how we keep drawing it, right? And then as I create, in light blue, my new build, my 3' end, we've always been moving towards the replication fork. And the reason then this is important is as that replication fork becomes unzipped with helicase, whip, right? This can keep going on and on and on and on and on until the end of the DNA strand, right? As long as there's space opening, this continues to be the 3' end. And we can continue to add on to it interrupted, right? Because it's still following our rule. It's an open 3' end, and we can always add on to it. So this is why we've been calling this continuous replication, right? We've always got that open, empty Lego bottom, so we can always snap new Legos onto it. This is pretty much how we've talked about this up to this point. So before we talk about anything else, does that make sense? Okay, so all we're doing is we're calling that the leading strand. That's the new piece we're adding here. So we're giving it a name. Right, because it can lead the charge. It can always move forward. Okay, I can hear voices, but I can't tell if you're just like commentating together, which is okay, or if you're asking a question. Okay, just wanted to double check. All right, so the lagging strand then would be if we consider, I'll put a yellow box up here, all of these guys on the other side. Okay. Now all our rules still apply here. So if we think about all parts of our replication bubble, we realize in some portions of the circle we're going to run into some trouble because of this only add to the 3' end business. Okay? So let's look at some of the guys in my yellow box. Okay. If I take the one that's furthest to the right, even if I'd gotten to start there, foot. Okay, I'm actually drawing. So there's my 5' end. It's fine. And I can only add on the 3' end. Even if I was able to do this in one go. Okay, I'm only going to be able to go up to the 5' end of the other piece. And I'm not going to mow that down. That's fine-ish, but I have one other problem. As helicase unzips in this direction, right? So if I unzip to the right here, how do I copy all of this top stretch up here, right? Because the problem we have, this is getting a little messy, so I'm going to move my yellow box, is listen, you rude little thing. I have a 5' cap right there, correct? So I can't just do what I did inside my black box. I can't just continuously tag pieces on, right? We can't add to a 5' end. So as we think about moving to the right here, that's the problem. Okay, that's the crux of our issue. Okay, we're stuck with a cap here. We know we still need to keep moving to the right. We unzipped it. Okay, but in order to do it, instead of just adding on nicely, because we have this 5' end, we got to start all over. So a new primase is going to have to come in, lay down a primer, a new 5' cap, so a new polymerase can come in, so we can do the one rule we know we have, and that's adding on a 3' end. Warp. Okay, that's fine until we unzip it some more. Okay? So what ends up happening is every time we unzip this to the right, we're faced with the same problem. Can't add to that 5' end, right? That cap is a cap. All right, so now I need to start a whole new segment with a whole new 5' cap, so I can then have a 3' end to add on to. Okay, so we've got two separate pieces here. Our leading strand has it easy. You start once, and then it's just like a nice day out in the English countryside where you're walking and go on straight forward. On our lagging strand end, man, it's like a relay race. You go for a few bits, then you stop. You have to go whip like a typewriter. Start at the beginning, go at the beginning, cha-ching, start all the way at the beginning, right? We only get to do teeny pieces. So this is what we mean by discontinuous. Okay, that all of the replication that still is happening in this direction is done in parts or discontinuous chunklets. Right? Because we always have to start a new piece, so we have a 3' end to add on to, right? Because we can't add. And rule number one, right? We can't add to that 5' end. So these chunklets that we've created, each individual one has a name. And they're called Okazaki Fragments, named after the gentleman that discovered them. Okay, with me so far, okay, why we have to do what we're doing, okay, and how it's happening. Okay, so all of this is occurring, and here we are again, okay? We just need to remember our rules. Okay, keep 5 opposite of 3, and we're only adding on that 3' end.
So are the Okazaki fragments those little spots that are circled in yellow, like little?
Correct. So she asked if the Okazaki fragments on here are the things that I've circled in yellow. So yes, those are examples of Okazaki fragments on our page. So anything that has become these discontinuous chunklets on the lagging strand, those are all called Okazaki fragments. It's a nice word for tiny chunklets that we can't just do in one long piece. Sounds much more sciencey, Okazaki fragments. So then we're left with one last issue, is we have chunklets, right? So even after, when we're all done, right, when DNA polymerase 1 goes back through, pulls out that red primer, and replaces it, I'm trying to color this in very poorly, with DNA, these were done in chunks. So there's going to be very small gaps in between them, right? Where the 3' end of the previous chunklet, or Okazaki fragment, didn't get to match up to the head of the next Okazaki fragment, right? Because DNA and RNA don't match up very well. So after we switch those out, we need to fill in these teeny weeny gaps between our fragments. So now we're going to add one more support staff. DNA ligase. And he's going to come through and put nucleotides, a handful, right, of DNA, the name implies, in between the gaps of our Okazaki fragments. So we get a nice, smooth, continuous set. Okay. So in the end, we still end up with a continuous stream, like the leading strand. It just takes us several steps to get there because we had to make it in pieces. I actually have a video for this, and the sound seems to be working in here today. So hooray. Kittens are cute. Obviously not my goal here. There it is. Apologize if this blares.
The other strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary
That's DNA polymerase 3,
to
right?
synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA.
This is DNA
Next,
polymerase
the
1.
enzyme DNA ligase links the Okazaki fragments to form the lagging strand.
And of course we haven't identified it, but who's the green guy in the corner? Helicase, good. And they did a very quick job of pointing out that kind of these clearish, I don't know if I'd call them blue, the clearish guys, of course, are our single-stranded binding proteins, right? Keeping everything apart.
The other
No. Shh. We're not playing that again. Okay. That is all of the pieces that I care that we know about for DNA replication. Okay. So you have a couple images at the end that summarize everything, a bit like our in-class activities do. So any questions about anything that we've done today? Or at any point?
So somebody asked in the chat, why are there leading and lagging strands on both sides in that picture?
And the one I have up here? Or here? I guess it doesn't matter, right? So remember, when we're doing this, right, DNA replication, woo! Just want my epic pen drawing. Is occurring on both parts, right, the top and the bottom strands, or parts of our replication bubble. Give. Okay, so as I think about this, okay, my top strand has a 5' and a 3' end, right? And my bottom strand has a complementary 3' and 5' end. Okay, so both of these strands will need to be replicated, right? Remember, when we think back to what we're doing here, right, all the way back to what's being done, our summary piece, this whole process was semi-conservative, so both these pieces will be pulled apart. Okay, they're the parent strands. Okay, so then our daughter strands will be made up of one parent strand, right, the OG, okay, and then one built mirrored strand. So all we're talking about for each of these is how do we build that mirrored daughter strand. That's what we're doing here. So that needs to happen for both. So if we just focus on the top strand, okay, I have a leading and a lagging strand as I put in a 5' end primer. Okay. Moving in my 3' direction. Stop touching me. Okay, remember my 3' end is going to be complementary. So, or opposite. So here's my 5' end. So my 3' end is going to move that way. That's continuous as I open this up. Just focusing on the top strand, right? That's going to be able to continuously add to that 3' end forever and always in all eternity. So that's my leading strand for the top. If I look to the other side of just the top, right? This is where I get my lagging strands in. So I put my 5' end in. Okay, I can still move to the left. That's still my 3' end. And I'll be able to go at least till I run into another piece. But remember, we can't move to the right. If I go to the right, then I would have a 3' end meeting a 3' end. Right, that's not complementary. That's not opposite. So we can't do that. So I'm going to go ahead and erase that. So the only way to make for the top, right, replication happen, continuing on to the right as we open that up, is to continuously create new 5' ends. Right, 5' and 3' are opposite, so that's okay to do. Making those lagging strands or Okazaki fragments. Okay, now I was just looking at the top, right? So now we need to look at the bottom, which also needs to be replicated. Okay, and it's going to have the exact same problems, okay, just on the opposites, right? Because everything is opposite. So I have my 5' here. So as I make my 5' end here, my leading strand, or my strand that's going to go continuously, right? I need my 3' end to be opposite of the parent strand 5' end. Like that. So this is my leading strand, my continuous strand. Okay, so that strand can proceed on the bottom, right, to the right for all eternity and forever. So it runs out of space. But as we look to the left, we see we have the same problem we had before. As I put a 5' end in, if I were to go to the left, right, I'll have a 3' with a 3'. So I can't do that, right? That's not opposite. It breaks our rules of complementary. So I need to go to the right, which is fine. I'll go till I run into something else. But as my replication bubble opens up to the left, we'll erase this because this is a rule breaker. We can't do this. Once again, I'll find myself needing to make new 5' ends. 5 and 3' can be together. Right, that's good with our rules of complementary. And so we'll again end up making those Okazaki fragments and a lagging strand. So we almost want to think about treating each strand, right, or each half of the replication bubble as independent of each other. Okay, because ultimately they will be two separate daughter strands. Did that clearly answer your question? I can't see the chat, so I'm going to go with...
So somebody else asked, how far does it open before the new Okazaki fragments start? And are the fragments symmetrical?
So the answer is it depends how far open it goes, but not very. It's pretty quick. So as helicase is opening the replication bubble, for both the leading and the lagging strand, these are continuously adding. So we're getting pretty small chunklets for the most part of these Okazaki fragments, right? Because our polymerases work very hard. I'm not sure I understand the symmetrical question. But the simple answer is probably not, and it wouldn't need to be, because remember, we're going to remove all of those pieces out using DNA polymerase 1 and ligase. So it's all going to be one continuous strand of DNA nucleotides when we're done. And so you won't see any of these little chunklets when the whole product is finished. This is only a during. Does that answer your question?
Yes, thank you.
Okay, wonderful. Okay, that was a very big, very good question. So did I get all parts of those questions?
I think you were good.
Okay, any other questions then before we put DNA replication to sleep? All right. Good night, sweet prince. Okay, Jasmine, will you pause and restart the recording for me? All 3 minutes of it?
Yep.
So we're just, thank you so much.